Hello, everybody. My name is Andrea Marshall. I am a staff scientist at Vanderbilt University, as well as a new adjunct research assistant professor at Meharry Medical College. And my background is in neurobiology, looking at the role of protein degradation pathways in neuromuscular diseases. I then moved on to do my postdoc looking at, again, the effect of degradative pathways on skeletal muscle atrophy models, as well as the impact of aging on skeletal muscle recovery after injury. And as I was doing the second part of my postdoc at the University of Iowa, that's where I met my current PI, who at the time we were both postdocs, going in and out of the microscopy core. And so most of my background with microscopy is more based in the confocal level. But since joining the Hinton Laboratory, I have been introduced into many different EM techniques. And so today I'll be discussing more of a historical view, as well as a little bit of real life example into a current project that we are undergoing using correlative light and electron microscopy. So a brief outline, I'll briefly go over a history of both light and electron microscopies that was beautifully penned by Lucy Collinson and Paul Vertke. And after that, I'll be following up with uses for modern CLIM, considerations. I'll walk you through a real life example, and then conclude and acknowledge. So a little bit of early history of microscopy in general. The earliest, sorry, I get nervous. But the earliest uses of lenses or things to magnify are date back prior to 300 BCE in the form of Nimrod lenses or reading stones. And those were usually either clear stones such as quartz or beryl that were then thinned and shaped until the desired result. People also used glass globes that were filled with water, as well as magnifying devices. And so major breakthroughs in what would become the first microscopes really occurred in the early 1600s. And so in 1616, Cornelius Drebbel invented the two lens or the double lens microscope. Around 1658, we had Kircher, who was a German monk. He first recorded and reported a biological sample that was viewed through a simple microscope. And in 1673, we had van Leeuwenhoek, who first recorded free moving structures in biological samples. And the term cell was actually coined by Robert Hooke in 1665 as he was examining cork bark underneath a microscope. And so the next major level of advances in microscopy occurred in the 1880s. And this is when, with the advances and improvements of microscope design and lens development, subcellular structures could be seen. And this was pioneered by Ernst Abbe, August Kohler, and Carl Zeiss, which is a name that many of us can recognize today. And so as these advances in microscope components were made, there were also advances in physics that led to the development of the electron microscope. And so here we have Louis de Broglie, forgive my pronunciation, and actually as his graduate thesis, he was inspired by some of the theories postulated by Albert Einstein and wondered what the form of electrons do. Do they follow a wave nature? And so as part of his PhD work, that's what he mainly described, and that actually got him the Nobel Prize in 1929. And so even though he wasn't really interested in the application of that knowledge, there were many people at the time who were interested in that application. So physicists such as Martin, who was in Brussels, Ernst Ruska and Max Knoll, who were in Berlin, as well as Ernst Bruch, who was also in Berlin, independently developed TEM prototypes. And so really the first recorded instances of CLIM did not occur until the 1960s and 70s as technologies continued to advance, which allowed for higher resolutions and better sample preparation. So the first instances typically focus on using similar but separate samples independently imaged by light microscopy and EM as a way of proof of concept that you can gain different knowledge from the different imaging modalities. These then led to advancements where people use the same samples, such as a tissue biopsy, and adjacent sections were prepared for either light microscopy and EM. But again, at this time, the results from these findings were typically reported in separate publications. And so that then progressed to more of what we consider the modern term for CLEM, which is using EM to improve the resolution of light microscopy samples within the same sample. And so these images that you see were taken from that publication with the first reported instance. And what he was looking at are blood samples taken from rabbits. And he first imaged them in light microscopy and then used electron microscopy to actually view the topology and the changes that occurred in the blood cells. And so here I'm just going to discuss a few select and very brief breakthroughs that have allowed to what we consider the modern revolution of CLEM. And so again, in the 1930s, you had the development of transmission electron microscopy. In the 40s, you had the co-development of the scanning electron microscopy, as well as the birth of what we know as modern immunofluorescence by Kunz and colleagues. In the 40s and 50s, you also see the development of ultramicrotomy, which allowed for ultra-thin sectioning and better sample preparation. You also see in the 1950s the development of confocal microscopy, which then all collectively led to the development of CLEM in the 1960s. And so what we see with each decade, as there are advancements in technology, material uses, pioneers in chemistry or other things that make different stainings and different natural resources possible, you see the explosion really of technology. So in the 70s, you see focused ion beam being invented, as well as improvements in laser and computer technology that allowed for scanning laser confocal microscopy. And really, as you see in the 80s and 90s, you have an explosion. So at this time as well, you see an expansion in antibody hybridoma, or so the ability to specifically label proteins of interest. You have developments in genetic technologies that allow for gene sequencing and targeting of specific genes of interest, as well as knockout, knockdown, overexpression technologies that were developed that really allowed scientists to ask more nuanced and finer-tuned questions. And during this time as well, you see development and explosion of the identification and cloning of fluorescent proteins, the development of fluorescent probes, as well as fluorescent dyes that allowed for different levels of specificity, which really are the powerful element of CLEM. And as well, in the late 2010s, you have the expansion and development of volume EM techniques, as well as 3D EM techniques, invention of AI and development of AI, as well as 3D modeling and expansion of analysis tools, which really allows, the world is at your fingertips of what you want to discover. So again, what you see here is that as times expanded, particularly from the late 90s into the late 2010s, you see that there is an expansion in the number of publications that mention CLEM in some form, as well as an increase in the citation. So this is a very burgeoning and growing field. So modern CLEM, what are some good uses, and why go through the trouble of extra steps of using multiple imaging modalities? So brief sections that I'll cover are just the ability to selection. There's with a lot of genetic techniques, you have only a subpopulation of cells of interest or that contain your modification, or there are only certain cells, the responses in certain cells that you're interested in. And so CLEM allows for you to selectively isolate those cells for further examination. It also, EM in general is a static technique. And so one of the beautiful powers of CLIM is that it allows you to add in more dynamic responses as well as time lapse into your questions, as well as with the limitations of basic light microscopy, EM is still the standard for certain localization of your proteins of interest or organelles of interest. And so this is an image from actually one of our data sets where we are interested in looking at how aging impacts mitochondrial morphology. So although we're able to identify mitochondria readily, as well as identify neuronal axons by this thick electron density around the myelin sheath, we are not able to identify what cell type we're in. And so use of CLIM would be a very powerful tool to help us fully understand the consequences to cell type specific mitochondria within the hypothalamus and the amygdala, which are two regions of interest for our research based on their roles in blood pressure regulation and neurodegenerative disorders. So taking it back, old school, I have done a ton of fiber typing. So the image that you see on the far left is fiber typing of mouse gastroc. And you see a combination of three muscles, the gastrocnemius, both the medial and the lateral heads, the plantaris. You see the nerve that goes through that bundle, as well as the part of the soleus. And so really one of the things that CLIM would allow if one is studying muscle and there is a fiber type specific expression pattern in your gene of interest, or if the type of fiber, which they do have different mitochondrial counts and there are different responsiveness and properties of those fibers are important to your biological question, CLIM is a powerful tool that will allow you to selectively show that the changes of your interest occur in the correct cell type. And what we see as well, which are images adapted from Chris Adams' nutrient-sensitive paper looking at the role of ATF4 in skeletal muscle atrophy. And in these cases, live animals were electroporated with either GFP or ATF4 overexpressing plasmids. And you can see readily the effect of elevations of ATF4 on muscle fiber diameter. However, if one wanted to explore the structure of mitochondria in these specific muscle fibers, as you can see, GFP is not expressed in every fiber type. So a tool like CLIM would allow us to specifically analyze fibers that either contain our gene modification of interest or are void of those interests. And we can compare the effects of mitochondria morphology in each. As well as, again, we have many neural projects that are starting in the lab that we're very interested in changes within a specific subpopulation of neurons, which are known to regulate different elements of blood pressure regulation, stress response to stress, as well as overall metabolic regulation. And so a tool like CLIM will allow us to selectively look at mitochondrial changes, again, because we're a mitochondrial lab, in that subpopulation with our gene manipulation and directly correlate how the mitochondrial changes contribute to the overall physiological phenotype that we see. So another powerful element that CLIM allows us to look at is that of time. Again, EM techniques are static in nature. And so when you can pair those EM techniques, I'm not sure if the videos are wanting to play or not, with different confocal modalities that allow you to look at real-time responses to either a drug treatment, a gene knockout, knockdown modification, or if you want to observe whether or not when you modify your gene of interest, you have changes in association or localization of different organelles. CLIM is a powerful tool for that. As well as there are many dyes that can actually report different functional changes. So if you paired your imaging with, say, a pH sensor or a calcium dye sensor and do your modifications or tests, CLIM is a way that you can correlate those functional changes now with the morphological changes that you see in EM with ultrastructure. And then last but not least, CLIM is also a good way to really confirm localization. Again, even though the resolution of light microscopy is vastly improved, EM is still the traditional or standard way to prove localization of your protein or chemical of interest. So here is a clip from a paper where they were looking at expression of DAG lipid and localize them to specific subcellular compartments. So how would one start? So the first thing when you're starting a project like this is you really need to consider what is the question that you're trying to answer. With all the vast modes of light microscopy that's available, as well as the vast modes of EM microscopy that's available, you really need to understand what it is you actually want to answer, and then determine is super resolution or ultrastructure really required for it, and what mode would be best suited for your tissue or your cell or tissue type. So what resolution is required? So how big is your sample? Are you dealing with whole human brain slices? Are you dealing with cells? Also, what's the best time to view? So is your phenomena of interest really time-based? So do you have to, say, induce your inhibitor and then 8 hours later you have either the expression of your gene or your changes that you're looking for? And so that's important to know, because you want to make sure that you capture that appropriate time frame. And when you get to the EM portion of CLIM, you're going to be examining your tissues a nanometer at a time. So you want to make sure very well that your target phenomena is present. And then also other considerations with that is how abundant is your phenomenon. So if you're looking at, say, a whole muscle slice and you're interested in immune cell infiltration, those cells are very small and the muscle is very large. And so using a technique like CLIM will help you isolate the specific areas of interest. And then also, how many observations are required for you to draw a conclusion that actually addresses your question? Other considerations, is room temp, EM, and imaging okay, or do you require cryo? And so one of the things during the EM process is many of the fixatives as well as the resin embedding process can create distortions in your tissue as well as in your proteins of interest. So if your question requires a nascent confirmation of your protein or organelle of interest, then cryo may be the way for you to go. As well as another huge consideration is, what do you actually have access to, and how are you going to get your samples there? And then as well with each of those things, what are the limitations of those technologies? And then once you have all these beautiful images in both light microscopy and EM, how are you going to analyze those data, and how are you going to correlate them? And the more work that you can put out in this thought process early with your study design, the higher likelihood you'll have a success. And shout out to all of our people who run cores. Your very best resources, in my opinion, are your imaging core personnel. They have years of experience. They have familiarity with all the equipment that is present in their core. They've seen potentially many projects like yours, or people who have asked similar questions and can give you very good advice. Also, they can give you feasibility. So is will it take 3 weeks of milling for you to be able to find what you're looking at? Or if you're looking for like a single molecule, like a needle in the haystack, is it really worth the time and effort it takes to use these techniques for your project? So the better you can understand and explain your question and your needs, the better they can help you. And definitely advise your study before you start, because there are many considerations. Again, as you're prepping your sample, they have to be amenable to both the light microscopy mode that you choose as well as EM. So you don't want to choose a light microscopy technique that will obliterate the ultrastructure of your sample by the time you get to EM. And here pictured are the key core personnel at Vanderbilt Scissor Core, and I have gotten phenomenal help from absolutely everybody. And so here I wanted to just briefly, with the time that I have left, walk you through what it's like doing a CLIM experiment. So here we're interested, so with a collaborator, we're interested in understanding the role of a specific post-translational modification on the association of mitochondria with the ER. So for this project, we decided to use a cell model, and we are overexpressing the enzyme that's responsible for adding this post-translational modification through use of a viral vector. So we chose super resolution imaging microscopy because the organelles that we are interested in are very tiny, so the mitochondria and the ER. Dynamics were not important to what we were going to do, so we chose to do fix the samples and use ARIScan imaging. So how do we actually see our structures of interest? So we know either our control or our cells of interest have either the M-Cherry labeled control vector or enzyme, M-Cherry labeled enzyme. So we're looking for red cells. We also have mitochondria that are labeled with an EGFP, and our ER was labeled with BFP-KDEL. And how do we track our cells of interest? We used Matec gridded dishes so that we can actually localize our cells of interest. And here is a picture using DIC of what that grid looks like and what the cells look like. And the image that you see below is when you do not have the right polarization. So if you stick your dish onto the microscope and it's not at the right angle, you will see almost nothing. So it took us like an hour to figure that out the first time we were doing it. So we received our samples. They were plated on a Matec gridded dish. What I just said, those grid lines are invisible unless you have it at the right angle. And although our collaborator gave us their cells of interest, we went back and we imaged our cells because one of the things they did not know to consider is that any cell that falls on a grid line or on one of the either letter or number labels is oftentimes will not be available in the EM when it comes time to milling. So we chose cells that were in the similar region, that were M-Cherry positive, that had a good GFP and BFP signal, that were in the original region of interest, but many of them were not the same cells. The other thing that we use this technique for is that there were many cells that were going through various levels of apoptosis or were actually dead. So we used the DIC as a discrimination thing or inclusion-exclusion criteria for the cells to carry through the rest of the project. And so again, we had a very big dish. There were very many cells, but we have to consider what is available or how large of a platform we have in the EM modality. So we chose to cluster our cells of interest within one quadrant so that after the resin embedding process and sectioning of the dish, that we would be able to identify our cells of interest or that they were close and be in the same block when we got to the EM. And so here is just one of the images from a control cell using the 63X on the ARIScan. And so you can see the cell was M-Cherry positive. We have a beautiful mitochondrial signal as well as a wonderful ER signal. And that's it merged. Another key consideration is note-taking is your friend. So for each of these groups, we took into account what quadrant in the grid it was. We took specific notes on the cell of interest so that someone could go back and identify if it was in a cluster, if it is in a cluster, is it the top left, bottom right, where it was located. We also kept just points where we could follow along in the process of this the whole process. So did we take our 10x image? Did we overlap and circle exactly which cell? Make sure we took our super resolution. Did we process our super resolution? And what day and time we did everything. So the next step was our EM processing. We chose to proceed with FibSim. One of the things to note with FibSim is a lot of times you get thousands of images and it can take hours to days to mill. And you have set cool down times with this. So with that, those things in mind, we debated time expense with the resolution that we need to see mitochondria and ER contacts and chose to proceed with 6 voxels. We embedded the resin, or we resin embedded our samples. We also used the fib to make etches in the resin just in case when the dish was popped off, if the grid lines did not leave a groove in the resin, we would still be able to find our cells of interest. We targeted 3 cells per group, 2 regions of interest per cell to proceed. And hopefully my GIF is playing, but this is just showing you the milling that we got from one of the samples from FIPSIM. So then we milled all our samples. We went back to our confocal. So to do our overlays, some of the considerations that we had to take into account is reconcile the shrinkage associated with the EM resin embedding process, as well as the orientation. So you're imaging from different angles between your light microscopy and your EM microscopy, and you need to take that into account. And one of the ways that one of the tricks that we used to be able to do this, as well as using different programs and stuff, was to visually identify specific landmarks to help us correlate the right areas. And so once we got those images, the next thing we have to do, what are we going to do with the volumes? So we're currently in the process of trying out different 3D image analysis programs such as Dragonfly as well as Napari. And we are training the models to be able to identify both mitochondria as well as ER membranes so that we can do our comparison. And this is Oleg Kovtun, who has been a phenomenal help with that. And so my conclusions, microscopy is advancing at the speed of light. And so soon, the only thing that will be our limiting factor is our imagination. CLIM offers many different levels of specificity, localization, dynamics, and resolutions that each modality alone don't offer. Some of the limitations are resource access. So what do you have available for you? A bonus and a limitation is that there are broad ways to this approach. There are so many different types of light microscopy and EM microscopy that you can use. So it could be a bit daunting for starting. But that's why I highly recommend going to your technical resources. And then the data that is generated from this type of technique is very large. So you requires a high computing output as well as high memory. And it is still expensive and it is still time consuming. And with that, I would like to thank everybody and thank our lab. And I will entertain questions if we still have time.